just put you down on the screen. So thank you very much. I'm very pleased to be there and to present my work on carcinogen and what is the definition of a real carcinogen. So I'm a toxicologist, so I do experiments on biology, and for a few years now, I try to develop a conceptual activity about toxicology. So I'm going to present my work in progress today. So firstly, I want to thank my founding and in particular the INRA and the Institut National of Cancer that found my research, and I declare that I have no conflict of interest. I also thank my team in Toulouse in Toxalim lab with many toxicologists that do experiment on pollutants and the origin of cancer, and also in Rennes and in Paris. So I also thank my collaborators in human and social sciences. As you will see, my topic is not only focused on biology, and I work with economists, sociologists, historians of science, agronomists, and this is why I'm here today. I work with Anouk Barberousse in philosophy of science and more particularly in biology. So I will present our work that we have done together. So firstly, to give you a definition of a carcinogen, if we simply look at the biologic definition that is also written in Wikipedia, a carcinogen is any substance, radionuclide or radiation that promotes carcinogenesis, and carcinogenesis is a process that is the formation of cancer. So in environmental carcinogenesis, we look at the effect of all the things that can be outside intrinsic factor. So it can be a natural exhaust, also plastics, tobacco, radiation, all the things that are around us. And just to give you an idea, we estimate that 40% of cancer would be preventable by changing our way of life. So it's like tobacco, alcohol, diet, and physical activities. And we estimate that 5 to 10% of cancer are due to environmental factors, so directly with pollution, radon, UV, or pesticides. So it's based on epidemiological studies. So today my presentation will focus on the definition of carcinogen and to learn how sometimes science can be surprising, and you will see. Anyway, so my first part will deal with cancer and the environmental knowledge that we have. More particularly, I will focus on pesticides. And finally, I will present the gaps that exist between basic biology and regulatory definition of carcinogens. So firstly, the first link between cancer and environment were described by Rachel Carson in 1962 in a book that is The Silent Spring. And she performed the first work on the impact of chemical on environment and health. And it's also the first work describing the potential links between chemicals and cancer. So the link between cancer and the environment was quite clear, and it was at the beginning of the 60s. But actually, when we look at the history of science, we observe that the research on the origin of cancer really focused on the mutation and the genetic origins. And it was particularly explained by the work of Matthias Girel that noted that actually the founding and the orientation of cancer research with the founding of the Human Genome Project create a field of ignorance about the knowledge that we can have between cancer and environment. And we should also put it in the context of industrial development with the the idea that chemicals improve health and progress for agriculture and also manufacture products. So for the science and the position that it was chosen, actually, maybe the influence of the chemical industry to put forces on research on cancer and genetics was voluntary or not, but it was a good way to not perform experiments on the link between environment and cancer. And it was partly well described by Robert Proctor about the tobacco lobbies that finance the research on the origin of genetic origin of a lung cancer. So it's something that we think we should think about and say, finally, now we are in 2020 and we have still the doubt about the link between cancer and environment. So it's something that can be surprising. So also another story about the controversy. Maybe you learn about this work about Tomasetti and Hovelstein in 2015. Because this team described by statistical and mathematical models that mainly cancer was due to bad luck. And bad luck, it was behind the fact that the division of stem cells and the cancer incidence was really due to DNA replication error, and that the impact of environment could be very weak. And actually other studies create a controversy in science, and 2 years after that, they propose a new model, and they differentiate the hereditary factor, the replicative factors that are related to the DNA errors, and for some cancer, the environmental effects that could be linked to some very particular cancer that is lung, thyroid, intestine, and pancreas, for example. So now if we look at the knowledge that we have about environmental carcinogenesis, actually it was described in the 18th centuries by Percival Pott, who identified the first occupational cancer in chimney sweep. Actually, these children was affected by scrotum carcinoma, and it was due to the presence of polycyclic aromatic hydrocarbons in soot. So now I'm going to introduce some chemicals, molecules, but don't be afraid. It's just polycyclic aromatic hydrocarbons, and they are described as mutagen and carcinogenesis, with the ability to form DNA adduct and to be genotoxic. And these compounds were classified as carcinogen type 1. So I will describe you the mechanisms when a cell is exposed to hydrocarbon. These molecules is very lipophilic, and the cells will try to eliminate it, so these molecules will be oxidized. And the oxidation of these molecules fatally produce a molecule that is very reactive with DNA. So here you have DNA structure, and here it's this metabolite that can be inserted into the basis of DNA and modify, so introduce mutation and so be genotoxic. So here we have the idea that carcinogens is an agent that is able to damage DNA. So it's really linked with the idea that cancer is a genetic disease. But when we look at all the way and all the hallmarks of a cancer cell that was presented by Hannah Hahn in 2011, you see that so many processes are involved in the formation of cancer cells. So here we have genome instability, mutation, but cancer cells can also resist regarding cell death, dysregulate metabolism, energetic metabolism, proliferate a lot, evade to growth suppressor, avoid high immune system, They have immortality at the permanent persistent replication. They also induce inflammation, activate invasion and metastasis, and promote angiogenesis. And when we look at the effect of these polycyclic aromatic hydrocarbons, as I described you, they are genotoxic, but they also modify all these hallmarks that create that transform normal cells into cancer cells. They also induce particular mechanisms that are signaling disruption. Actually, these compounds have a specific nuclear ligand in cells, and they also can have endocrine disruption effect. So just to give you this slide, actually as a toxicology, I work on contaminants and carcinogenesis by considering different chemicals, so that I call old and persistent chemicals like polycyclic aromatic hydrocarbons, also dioxine. I used to work on diet and neoform compounds during digestion that could be carcinogenic also. Now I will present some results about pesticide and more particularly some fungicide. To give you an idea, when for plants they are exposed to fungi that can eat them and decrease the development of plants. So we use fungicide to kill fungi. So today I will present you some pesticides, fungicides that are succinate dehydrogenase inhibitor. And so I will present you here, it is a mitochondria, that is the site that produce energy with ATP and consumption of oxygen. And the complex 2 is involved in the respiratory chain. And this is the target of this pesticide. So here we have the respiratory chain that is coupled with the electron transport. And the complex 2 is the succinate dehydrogenase SDH here, and it's a complex 2 that is also involved in the Krebs cycle. So it's a cycle that allows the consumption of succinate and in transformation into fumarate. So this pesticide, so target complex 2 in the mitochondria of fungi, but as it is a highly conserved function, we have some evidence that they also target human enzyme and also enzyme of many living beings. In parallel to this knowledge, there are also human clinical diseases that were described, and it was related to some mutation in the SDH. So this enzyme is mutated, so in different subunits, subunit A, B, that led to neuropathies and encephalomyopathies, that is the Leigh syndrome. So there are also mutations in other subunits that trigger cancer, like paraganglioma, stromal tumor or gastrointestinal tractus, and pheochromocytoma. So for the 1995, we know that with some people that have mutation in SDH have a lower activity of this enzyme, and they have some disease related to this mutation. So as a toxicologist, when we discover actually that the chemical inhibition of SDH can occur when we are exposed to this pesticide, we have knowledge in biology that can predict the incidence of the succinate dehydrogenase inhibition in humans. So the mechanisms by which the inhibition of SDH trigger this pathology that I described is that they induce metabolic reprogramming. So you disturb respiration, we disturb Krebs cycle, so it induces metabolic reprogramming, and also the accumulation of succinate because the enzyme is stopped. induce epigenetic modification. And for epigenetic modification, it will actually modify the unrolling of the DNA. So it's not directly mutation of DNA, it's not going to change the sequence, but it's going to change the accessibility of genes to transcription and expression. And actually, these two criteria of carcinogenicity, the process that can trigger carcinogenesis, are not taken into account by the regulatory agencies that evaluate the carcinogenicity of pesticides. So now I will just present you the gap that exists between the knowledge that we have in cancer biology and the regulatory toxicology that actually allowed some compound that can trigger cancer with different mechanisms that are not taken into account. And we have also the expertise of the WHO, the World Health Organization, and the hierarchs that I will present you just right now. So for the pesticide, you know, they are evaluated by different agencies. So I will present the work of the agency that is the International Agency of Research on Cancer that was created in the 1965. And it is an intergovernmental agency from part in the World Health Organization of the United Nations. So it's not a regulatory agency, it's just an agency in the service of public health. And it conducts and coordinates research into the cause of cancer, and it collects, publishes surveillance data regarding the occurrence of cancer worldwide, and they work also on the writing of monographs that identify carcinogenic hazards and evaluate environmental causes of cancer. So they take the literature, the scientific literature, based on qualitative assessment of animal and human evidence. And according to the strength of evidence, they propose a classification. So when sometimes we have data on humans, that is epidemiological studies, experimental animals, so it's more toxicological studies, and also other relevant data like mechanistic studies on cells to understand how a compound can be carcinogenic. And according to the strength of evidence, if we have a lot of evidence, they classify into the group 1, probably carcinogenic to humans if they have sometimes good evidence in toxicology and barely less in humans. And also we can decline it with possibly carcinogenic. After group 3 is that we have not enough data to classify compounds, and group 4 is that it's probably not carcinogenic to humans. So this classification can evolve. If we increase the knowledge, we can reconsider the classification of a product. So we have this work of INRA that is not a regulatory agency, but what now concerning the regulatory agency, and more particularly the European Food Safety Authority. So it is the authority that give the authorization to the firm to make a product enter the market. And the work of these agencies is to evaluate the risk. So the risk is take into consideration the hazard that is based on mechanistic study and toxicological study, but also the probability to be exposed. So to give you an idea, it's like if you evaluate that a lion is very hazardous for us, but our probability to meet a lion today is very weak. So they can allow a hazardous product if we estimate that we will not be exposed to it. So the evaluation is based on scientific literature like INRA, but also it is based on data provided by industrials. And actually industrials provide data that are carefully chosen. Actually, they have some guidelines that are defined by the OCDE, so it's a commercial exchange, and they have to carry out some carcinogenic experiments to evaluate if the product that I want to sell is carcinogenic. And actually, when we look at the carcinogenic criteria, it's mainly based on genotoxicity evaluation. This evaluation is very important because actually when a product is classified as carcinogenic, it can't be authorized or it is banned if the product was already allowed or retired to the market. So the genotoxicity and carcinogen is the very important characterization, and you will see that that is why actually the definition of a carcinogen can be changed, because when we enter the definition of carcinogen into a community that not work on biology but are in the market spirits, they consider that some criteria are good or not. And for instance, the main criteria to characterize carcinogenicity is genotoxicity. So when we look at the science that is used by regulatory agency, let me introduce a very old scientist that was Paracelsus in the 16th century that worked on poisons and said that all things are poison or nothing is without poison, only the dose permits something not to be poisonous. So the idea is that a compounds, if we are exposed at a low dose, we have no toxicity. And actually the regulatory agency are really focused on this definition. That is why when we consider science and the knowledge that we have, we can emit some criticisms with the parameters that are not taken into account. So the formulation with co-formulant, actually when pesticides are used, they are mixed with some products that are not the active substance. So to give you an idea, glyphosate is the active substance, but it's sold as glyphosate, as a Roundup. So in Roundup, you have many compounds that allow the entry of glyphosate into the leaf. And these co-formulants are not evaluated into by the regulatory agency. So they are not evaluate the whole formulation, they just evaluate the active substance. Moreover, they do not take into account the low dose effect. Maybe you heard about the endocrine disruption. So compounds and some pesticides have endocrine disruption effect by mimic the hormone activation. So, you know, hormones are effective at very low concentration. So the idea that the dose makes the poison is completely wrong. Moreover, they do not take into account the cocktail effect. Each active substance is allowed independently, but imagine when you eat an apple, this apple was treated by pesticide 36 times. So you can have more than 10 pesticides on one apple, and theoretically each pesticide is safe, but we have no idea of the safety of the cocktails that are present directly in food. Moreover, they do not carry out experiments according to the windows of vulnerability, more particularly when people are exposed during pregnancy, of a fetus, during childhood, when you have all this importance of hormone and endocrine disruption, regulation. So if you have chemicals, it can interfere with this process. And the difficulty is the delay that can exist between the exposure and the disease outcome. And as you know, cancer was more related with aging disease. So to attribute the origin of cancer with an exposition to chemicals 20 years ago, it's very hard scientifically to get the real proof of evidence. So to give you an image, actually, if we consider how the pesticide risk assessment is done, it's done like if you wanted to evaluate tobacco, but just giving you 10 cigarettes for every day for 1 week and just try to predict the carcinogenic effect of tobacco. So it's very unsatisfying method to evaluate the carcinogenicity. So now if I back to my the pesticide that I show you about the inhibitor of succinate dehydrogenase, actually when we look at the experiments that were done in rodents, so it's an empirical approach, you give increasing dose of SDHI. Actually, when we look at the data that are in the regulatory files, we show that we discover that more than 8 SDHI are really able to trigger cancer in rodents, like thyroid, in the liver, in lung, and uterine, and also ovarian carcinoma. And actually, all these compounds, so they are written in black, induce cancer, but they are authorized. And for some case, when we look at the report, you know, it's a regulatory report, it's more than 300 pages of raw data and experiments that are not really well presented. And actually some countries propose to classify them as carcinogenic, and sometimes, you know, it's erased. So here, when it's written carcinogen 2 here and it's suppressed, it's because actually they decide, for example, fluopiram has some evidence to be classified as a carcinogen, but they decide it's not carcinogen. So it's made with many compounds related to this category of pesticides. So to give you, you say, okay, if we have data on rodents, why at the end these data are not taken into account or decide not to be considered? And actually it's a process of science washing by using science. So to give you an example, they justify some cancers that are activated by a particular process that could be specific to be activated in rodents. So to give you an example, I will present you the nuclear receptor CAR. So CAR is the nuclear receptor that regulates energetic metabolisms, cellular detoxification. And this compound, this nuclear receptor, is activated by some pharmaceutical drug like phenobarbital. So phenobarbital is an anti-anxiety drug that is used in humans. And when we, if you know, perform experiment on rodents, you give phenobarbital to rodents every day, and they develop hepatic tumors. But in the population, so in humans, this exposure, you know, the treatment with phenobarbital do not induce tumor in humans. So in this case, we have a discrepancy between rodents and humans that can make us characterize that the tumors observed in rodents are false positive, so are not relevant for human. But that is done based on this knowledge, very particular related to phenobarbital. Actually, regulatory agency use this argument to assess how pesticide can activate the CAR receptor. So they produce literature in science and peer-reviewed journals, and they are not looking at, you know, the inhibition of succinate dehydrogenase to see if it's induced succinate accumulation or metabolic reprogramming. No, they look at the ability to activate this nuclear receptor. And the idea is to say, okay, when we give rodents this pesticide, we have an activation of a CAR receptor. So the tumor that we observed is not relevant for humans. And actually, they did that for one SDHI here, but for many also. And when we look at the, here it's like a peer-reviewed journal, and it's founded by Elsevier. And we have the impression that it's a peer-reviewed journal, but actually when we look at the editorial board, you see that there are a high representation of films and all the demonstration of the activation of CAR receptor are published in this journal. We have also toxicological science from the Oxford University, and also the same with another pesticide that can activate nuclear receptor. They use it also in toxicology reports. So we have this production of literature in some bad quality journals that actually produce scientific-like literature. And it's very confusing. And moreover, when we look at the reviews that were done on the scientific literature to describe mode of action and this interest to use it to consider what is really relevant for humans, we have a lot of articles. And here I give you an idea. All these people are related to Industria, or I also work in a big university or in the regulatory agencies. And so they use some words that are, the language is very particular. They consider that is some outmoded, and science and society, it's not a really strict communication that are normally found in the scientific literature, but they say, okay, if you consider that you have tumors in rodents and you say that you cannot allow pesticide, it's outmoded science and it's not good science, so it will stop progress and the progress of society. So it's very disturbing when you are a scientist and work on biology to be confronted with this kind of reviews. And actually, when we work with sociologists, you say, okay, when you take the name, we discover that it was linked to a particular think tank, that is the International Life Science Institute. That is, when you look at the website, it's described as a world non-profit organization that put together scientists from industry, agencies, and university in order to, produce sounding science. And this think tank is related to agri-food, agrochemicals, health, and they pretend to defend common welfare and the preservation of environment. And so they have some task force that discuss about what are the real definition of carcinogen and what criteria proof should be considered or not. And they wrote some literature publication that, if you have naive, you have many reviews that say, okay, if you have, for example, CAR activation, it's an indication that you have failed positive cancer. So just to conclude, the criteria of carcinogenicity actually depends on the communities that produce results, and there is a real gap between the basic cancer biology and the regulatory toxicology. And at the end, the origin of the knowledge is really important, the consequence of on the definition and the demonstration of what is a carcinogen, and it's really influenced by the presence of film, and it's supported by think tanks that allow the interaction between film scientists and regulatory agencies. And also it infiltrates all the world organization. I don't mention that, but we can also find some experts of this think tanks that are in some scientific society. And they are also in the editorial board of journals. So at the end, it's very confusing because with the progress of science, you know, this basic biology actually is not confronted with good science, and the regulatory framework for the entry of the product into the market is barely in favor of the protection of health and the protection of biodiversity. Because you take a risk. If say, okay, the experiments that we have in rodents, we consider that when you have 10 pesticides, you consider that you have 8 false positive results in rodents. It's very disturbing. So I work on that and was on the way how the expertise can be improved and how we can propose a new framework to improve the regulation of pesticides. So thank you for your attention, and I'm happy to take your questions.
Thank you. Unfortunately, we have run out of time, but I'm going to take one question. So if there is someone, please use the raise hand function.
Yeah, I'll stop it. Yeah.
So I don't see any hands raised right now, but I would suggest that we all, I mean, those of you interested, we can move to the Gather application, try it out. And perhaps if you do have some questions, not just to Laurence, but also to Lucie or other questions, we can talk we can talk there. Otherwise, we take 30 minutes break and reconvene here again at 12.